Okay, good morning, everyone. Welcome back. Hope you and your families are doing well. And today is supposed to be a beautiful day outside, so maybe get outside and just breathe some fresh air at this time. Okay, so folks, everyone made it through the class marker lab exam on Monday. I was happy to see that. There were no major technical problems with that, so that's encouraging. Just to let you know that the class average on the lab exam was a 74%, which is about 10% higher than your first lab exam class average. So that's very encouraging. So as a group, you have done much better on lab exam 2 than you did on lab exam 1. So very nice job. At this point, I have reviewed all the exams online and made some grade adjustments for things like spelling errors and things like this. So if you haven't logged in to see your lab exam grade since yesterday afternoon, I would encourage you to do so because the grades are pretty much final at this point and they have been reviewed. If you do have a question about the exam, if you have a question about something, by all means, please reach out to me through email, and I'll try my best to address your concern. But please log in and check your grade out. They are finished at this point. Do you have any questions? Okay, so Pamela asked, do we go to class marker to see the grades? Yes, Pamela, please do that. I won't be sending out additional information unless there's a technical problem of some kind, but you can log back in the class marker and get your exam grade. Other questions? Okay, gang. So today we're going to finish our coverage of chapter 10. Next Wednesday, as you know, you are scheduled for lecture exam number 2. Okay, so that's a week away. We're going to finish chapter 10 today. On Monday, we're going to start chapter 12, I believe it is, on the nervous system. And then you will be taking your second lecture exam a week from today. Starting this Monday, you are 4 weeks away from the end of your semester. Starting Monday, we have 4 weeks of lecture left, followed by your final exam. So it's not too far away, folks, and keep that in the back of your mind that there is light at the end of the tunnel, that you are going to get through the course, and that things will start normalizing out in the real world again, and we can go out and actually enjoy life again. So do you have any questions about our schedule or anything else moving forward? Okay, not seeing anything. What we're going to do is go back to the PowerPoint drawing that I was drawing last time, where we were talking about the activity at a neuromuscular junction. Okay, gang, so bear with me just one moment. Okay, so at this time, you should be looking at a PowerPoint drawing of a neuromuscular junction. We were talking about this at the end of last class. And also the chat window is up. So if you have questions, feel free to type them into the chat window or unmute your microphone to talk. Okay, so let's recap. We were talking about a neuromuscular junction. From last time, we know that we have a neuron next to the sarcolemma, which is the plasma membrane of a muscle cell. And we said that last time this whole process begins with a nerve impulse that spreads down the neuron. The nerve impulse is an electrical signal, which is really a flow of ions that go back and forth across the plasma membrane of the neuron. This is highlighted in the drawing by this red jagged fin-like drawing here, the red color. As that happens, as the ions move back and forth across the plasma membrane, eventually they reach the amino acids in the voltage-gated channels, which are made of protein. Those ions will neutralize some of the charges on the amino acids of that protein, and when you do that, you change the shape of the protein. So the voltage-gated calcium channels open in response to the nerve impulse. Calcium ions, which are present in the synaptic fluid, will enter the neuron. When that happens, the calcium will trigger the exocytosis of acetylcholine contained in the synaptic vesicles. That acetylcholine will enter the synaptic fluid and it will float across to the sarcolemma, the plasma membrane of the muscle cell. When that acetylcholine binds to the acetylcholine receptor embedded in the plasma membrane of the muscle cell will generate its own electrical signal, which we call a muscle impulse. Okay. Gang, do you have any questions at this time before we continue? Okay. Is there a picture or graph of this in our book? There is some pictures that I can give you a hint about. Some of the physiology that we are talking about at this time can be best viewed on page 310, page 310, page 309, 310, 311, talk about the activities that are occurring at the neuromuscular junction. Okay, so in terms of the neuromuscular junction, figure 10.9 is probably your best bet at this point. Okay, but there will also be things that are important in figure 10.10 on page 311. Okay, so continuing forward here, what we want to do is add some additional content to the drawing. And so what I'd like to do is add some extensions off the sarcolemma that come into the inside or the internal regions of the muscle cell. These extensions, as you may be aware of from the reading and studying you've done, are called T-tubules. Okay, so these extensions are T-tubules. Okay. Now, I'm going to have to erase some of my notes on the screen from the previous class because I need more space to write. Obviously, you don't want to erase any of your own notes, but please know that I'm going to erase a few parts of my notes on the screen so that I have more space to write new notes. Okay, bear with me a moment here while I finish this. Okay, wonderful. Okay, gang. So take note that the T-tubules are also known as transverse tubules, and transverse tubules are what your book calls as invaginations of the plasma membrane. The T-tubules are actually inward extensions of the plasma membrane that go into the interior of the muscle cell. Okay, so please be aware of that. You can perhaps see the T-tubules best on a different picture in your textbook, and that would be page 298, figure 10.2. You can see the transverse tubules in that drawing of figure 10.2. So we're going to be going back and forth between a number of pictures here, and feel free to do that while you're at home. Okay, so let's continue here. So we now know about the T-tubules. The T-tubules are invaginations of the muscle cell plasma membrane, And in addition to that, if you're looking carefully at your book, there are, in addition to the T tubules, you'll notice that the T tubules kind of extend in, through, and around this material that's actually called the sarcoplasmic reticulum. Okay, the sarcoplasmic reticulum is a network of membrane. It's a network of membrane that is the muscle cell's version of smooth endoplasmic reticulum, okay? The sarcoplasmic reticulum is smooth endoplasmic reticulum. And even though I'm very, very bad at drawing, what's important for you to appreciate is that the sarcoplasmic reticulum is the location inside the muscle cell that stores calcium ions, okay? So I'm just going to put some dots in here to indicate calcium ions, Ca plus 2, okay? So in our drawing, we have two sources of calcium ions at this point. We had calcium ions in the synaptic fluid, which is outside the muscle cell, and it's those calcium ions that enter the neuron when the nerve impulse reaches the voltage-gated calcium channels. In addition to that, there is a separate reservoir of calcium ions that is maintained in the muscle cell inside the sarcoplasmic reticulum. Okay, so we have two different sources of calcium ions. Okay, getting back to our story here, when the acetylcholine binds to the acetylcholine receptor, okay, so when acetylcholine A binds to the acetylcholine receptor, and we recall from last time that it actually takes 2 molecules of acetylcholine to bind to the acetylcholine receptor. When 2 molecules of acetylcholine bind to the acetylcholine receptor, this allows for the entry of sodium ions into the muscle cell, which is the first step in creating a muscle electric signal, a muscle action potential. So what's going to happen is, when acetylcholine binds the acetylcholine receptor, we're going to get a new jagged signal that goes back and forth across the plasma membrane of the muscle fiber, just like it did with the plasma membrane of the neuron up above. Okay, so we now have a muscle action potential occurring at this time. Now this is going to spread laterally throughout the length of the sarcolemma. But in addition to that, because the T tubules are actually made of plasma membrane, that jagged sawtooth, okay, sawtooth is a word I've been looking for a while, that sawtooth is actually going to go back and forth down the membrane of the transverse tubules. Okay. All right, so note number 6 in your notes here is going to be the fact that the muscle action potential spreads down the T tubules. The muscle action potential spreads into the T tubules. Now, what you notice there, not necessarily by my drawing because I'm very poor at it, but as you look at your own textbook images on the different pages I've highlighted, 298 at this point, that the T tubules run down and through this sarcoplasmic reticulum network, which in your textbook on page 298 is actually colored orange, and in the drawing on your screen is colored green. So long story short, what's going to happen is that the muscle action potential goes through the membrane of the sarcoplasmic reticulum. And you can bet that, let's see, let me do this here. Embedded in the membrane of the sarcoplasmic reticulum are voltage-gated calcium channels. Okay, so just like we had voltage-gated calcium channels here in the neuron, we also have calcium channels here in the sarcoplasmic reticulum. Calcium channels. So when the muscle electrical signal goes down the T tubules, the calcium channels in the sarcoplasmic reticulum open up, and the calcium that's inside the sarcoplasmic reticulum leaves the sarcoplasmic reticulum, actually comes through here and dumps out into the cytoplasm. Okay, so we now have calcium that has left the sarcoplasmic reticulum and has entered the cytoplasm. So number 7, calcium is released from the sarcoplasmic reticulum. Okay, sorry that the drawing's not so good. Calcium is released from the sarcoplasmic reticulum. Okay, so let me cover this from start to finish very quickly so you can see how everything fits together. At a neuromuscular junction, for the nervous system to stimulate muscle contraction in a skeletal muscle cell, what has to happen is that an electrical signal spreads down a neuron, which opens the voltage-gated calcium channels in the neuron. Calcium from the synaptic fluid enters the neuron and triggers exocytosis of the acetylcholine neurotransmitter in the synaptic vesicles, dumping that material into the synaptic fluid. The acetylcholine floats across and binds to the acetylcholine receptors embedded in the plasma membrane of the muscle cell, the so-called sarcolemma. That generates a new muscle electrical signal called a muscle action potential, which runs down these invaginations called T tubules, which pass through the sarcoplasmic reticulum. When that happens, the electrical signal will change the shape of the calcium channels in the sarcoplasmic reticulum, causing the calcium to leave the sarcoplasmic reticulum and dump into the cytoplasm. Gang, before I go any further, do you have questions? Are things making sense at this time? Because it's a chain reaction. It's one thing has to happen, then the next thing has to happen, then the next thing has to happen. Is there anything I can help you with at this time? Okay, you're being very quiet, so I hope everything's okay. All right, let's continue here. All right. So, okay. It's difficult to give you an exact image. So I'm going to suggest either figure 10.4 on page 301, or perhaps figure 10.6 on page 305, or perhaps figure 1010 on page 311, depending on what you want to look at. You want to go back and forth so that you understand things. Okay, so at this point, what's going to happen next is the presence of the released calcium into the cytoplasm is actually going to be the chemical cue that triggers contraction of the skeletal muscle cell. Okay, calcium is the trigger. It's the chemical stimulus, if you will, inside the muscle cell that begins muscle contraction. Not so good. Calcium is the trigger that starts muscle contraction. Okay, So at this point, I think what I'll do is go back to the Microsoft Word document and use that to simply talk you through the process using your textbook as a visual guide. So before I leave the PowerPoint drawing, do you have any questions? Okay, let's go to the PowerPoint drawing. Excuse me, let's go to the Microsoft Word document. Okay, all right, so, okay. We are talking about calcium being released inside the muscle cell. And this triggers the muscle cell to contract. Okay, so that's where we are at this point in our conversation. Okay, now, if you are looking at figure 10.10, what's going to happen is that the calcium is going to trigger a process called the sliding filament model of contraction, which is, in some books, is simply referred to as the contraction cycle. So it is what it is. But what has to happen next? Let's see here. On my PowerPoint, I was at number 8. So now let's go to number 9. Number 9. Calcium has been released into the interior of the muscle cell. What happens next is calcium binds to a regulatory protein, which I put into your notes last time, called troponin. Okay, troponin can be seen on figure 10.10. It can be seen on figure 10.6. It can be seen on figure 10.7. It can be seen on figure 10.4. So you pick where you want to look at this. But calcium binds to troponin. Okay, calcium binds to troponin, and troponin is attached to another regulatory protein called tropomyosin. So calcium binds to troponin, which changes the shape of troponin. Okay. Now, because tropomyosin is attached to troponin, when troponin changes shape, so does tropomyosin. And when tropomyosin changes shape, it actually rotates a little bit and changes its position. Okay, so calcium binds to troponin, which changes the shape of troponin. Because tropomyosin is attached to troponin, tropomyosin also changes shape and position. Okay. Now, what's key here? Let me ask you through the chat window. A question coming through the chat window. What regulatory protein blocks the myosin attachment sites on actin? Which regulatory protein blocks the myosin attachment sites on actin? From your study, from your interpretation of the book, what regulatory protein typically blocks the binding sites on actin that myosin wants? Go ahead and type in your answer into the chat window. ADP, thank you for participating, Suzette. That is not correct. Okay, Pamela suggests tropomyosin. That is correct. Okay, at least on figure 10.4 on page 301, you see this darkish yellow brownish band of protein called tropomyosin, and it is actually blocking the attachment sites on actin that myosin would like. So when tropomyosin shifts position, it exposes those binding sites. Okay, so number 9, once again, calcium binds to troponin, which changes the shape of troponin. because tropomyosin is attached to troponin, tropomyosin also changes shape and position, which exposes, meaning it opens the binding sites on actin that myosin needs to attach to. Okay. All right, folks, is that making sense? Okay, I hope so. You let me know what you need help with. All right, so now it's the calcium that pulls the regulatory proteins off their position, allowing for the possibility of a muscle contraction to begin. So calcium is the trigger that removes the regulatory proteins so the contraction cycle itself can work. So at this point, we move to figure 10.6 on page 305 and continue our discussion. So at this point, calcium has removed the regulatory proteins. They're out of the way. They're not going to block the process anymore. So at step 10 in your notes, I'm going to be starting with step 1 on page 305. And here we talk about the contraction cycle. Okay, so contraction cycle, I'll use the language of your book. Step 1, the blue ball number 1 on figure 10.6, page 305. The myosin head actually breaks down ATP, and that, of course, releases energy. So the myosin head can move into position to do what it needs to do. The myosin head breaks down ATP, which releases energy to reposition the myosin head. Okay, notice that this whole process is described as a cycle. That's very important that you appreciate it, okay? In other words, what we're about to describe is a series of events that repeats themselves over and over during a muscle contraction. So the myosin head breaks down ATP, releases energy to reposition the myosin head. Okay, that's step 1. Step 2, using the numbering of your own textbook, the myosin head actually grabs onto actin. Myosin head physically moves and touches actin, attaches to it, grabs it, binds it. The myosin head physically binds the actin, and when this happens, this is called a cross bridge. Okay? When the myosin is attached to actin, we have formed a cross bridge. Okay, so when myosin is attached, we have a cross bridge. That is step 2. At this point, what's going to happen is you notice between steps 2 and 3 in your textbook, there's a phosphate atom that's going to be ejected from the cross bridge, and that's going to help power the motion of the myosin head in towards the center of the sarcomere, which your book is trying to show you in step 3. Step 3, the myosin head pivots, pulling actin along with itself towards the center of the sarcomere. This is referred to as the power stroke step. Myosin head pulls actin toward the center of the sarcomere. Okay, this is referred to as a power stroke. Okay, and it's this step where the muscle fiber, the muscle cell, actually shortens. This is the step that physically shortens the muscle cell. Okay. So we have physically shortened the muscle cell at this point. And the next thing that has to happen in part of our cyclical process here, okay, is that the myosin head is going to have to detach from the actin so that it can relink and begin a new contraction cycle. When you think about this entire process, the thing that, an analogy that works a little bit for me, I saw your question, Pam, is that you should think about a rowing team. Think about a bunch of gals in a rowboat, and they each have an oar, and they're putting their oars into the water, and they pull back to push against the water to propel the boat forward. As they do that, they bring their oar back up out of the water, push the oar forward, and then push it down back into the water. So you have this cyclical process of oars entering the water, coming out of the water, going forward, and going back down into the water. So this process, this cycle, has to continue indefinitely for muscle contraction to continue. Okay, so Pam asked for number 3 to be explained again. Okay, during step 3 on page 205, the book says that the myosin head pivots, pulling the thin filament, okay, which is actin, past the thick filament, which is myosin. So the myosin head moves towards the center of the sarcomere, and it drags actin along for the ride. So you get an increase in overlap between the thick and thin filaments. That increase in overlap between the thick and thin filaments is what actually shortens the muscle cell. Okay. Pam, does that work? Is there something else that you need me to address? Okay. All right. So step 3, we've shortened the muscle cell. Now we're going to complete our cyclical process at step 4 with the myosin head detaching from actin. This is only possible because another molecule of ATP floats in and binds to the myosin head. Okay, so myosin detaches from actin when a new, meaning a new fresh molecule of ATP attaches to the myosin head. Okay. So, question coming through the chat window. Where is ATP coming from? Where's ATP coming from? Right? I mean, your book is assuming that you appreciate that. Your book is assuming that you know what ATP is, and it's assuming you appreciate where ATP is coming from. Okay. It's not calcium, no. Where's the ATP coming from? Okay, Lindsay says the mitochondria, and that's a decent answer. What I'm trying to get you to see, folks, is ATP, which is typically made in the mitochondrion by aerobic cellular respiration, is simply floating around in the cytoplasm. You have a whole bunch of ATP that's floating around the inside of the muscle cell. It's floating around inside all muscles, all cells of any kind, okay? So the muscle cell has access to an abundance of ATP. For example, if I told you to reach up your hand wherever you're seated at this time and grab a whole bunch of oxygen, you could do that, right? Because there's oxygen in the air that is in the room you're at. There's oxygen everywhere. Yes, it's too tiny for you to see it, but it is everywhere. ATP in the cell is the same thing. It is everywhere. So the ATP is simply present in the cytoplasm in a great abundance because it was made by the mitochondria. So this ATP, which is present everywhere in the cytoplasm, floats in, attaches to the myosin head, changes the shape of the myosin head so it releases its grip on actin, and it falls away from actin. It detaches actin. That's step 4. And then for the ore to go back in the water, to use our analogy, for the ore to go back in the water, we've got to push it forward first. We've got to reposition the ore. And that's going to bring us back to step one, where that molecule of ATP is broken. The energy released is used to reposition the myosin head. It's used to reposition our ore in the rowboat. And then we can begin step two, which is to attach to the actin and start the process again. So we go step 1, 2, 3, 4, and then we repeat. It's like washing your hair with shampoo. Rinse, lather, repeat. Rinse, lather, repeat. We do these steps over and over again. All right. Questions at this time, folks? What's not clear? Okay. Is there any aspect of neuromuscular junction activity or the sliding filament muscle, the contraction cycle that we've just put into your notes that you would like to talk about, that you would like to ask a question about? Okay, folks are awfully quiet. Okay, so hopefully that you're okay with that. And so we will step away from that. You know, it was probably 1 or 2 class periods ago that I gave you that flow chart connecting muscle contraction to bone movement. So I'm not going to type that back into your notes, but let's think about what's happening here. At step 10, we've gone through this cyclical process, which leads to a contraction of the muscle cell. Okay? So we put that in the flow chart in your notes. We started that flow chart with the sarcomere shortening, okay, which led to the myofibril shortening. Okay, this is all possible by the contraction cycle we just put into your notes, which then causes the muscle cell to shorten. When the muscle cell shortens, it tugs on the connective tissue layer, the endomysium around the muscle cells, which then tugs on the paramysium around groups of muscle cells we call fascicles, which then tugs on the epimysium, which is around the entire muscle itself, which then is connected to the tendon. So we tug on the tendon, which is connected to the bone from the periosteum. The periosteum is attached to the surface of the compact bone through the Sharpey's fibers. And finally, the actual bone physically moves. So there's a tremendous number of things that have to happen one step after the next at the molecular level in order to simply move your arm. Gang, do you have any questions? All right. If there's no questions, then what I'll turn to is a discussion of ATP production in muscle fibers. Okay, so energy production is talked about on page 312, 313, 314. Okay. Okay, so we're switching gears now away from neuromuscular junctions and actual contraction physiology to just make a few statements about the production of energy. Production of ATP in muscle fibers. Okay. All right. Most cells of your body actually have 2 different ways that they can manufacture ATP. Typically, those processes are called aerobic cellular respiration ATP production inside all cells can be made through two mechanisms: aerobic cellular respiration, which, if we're being honest, we did not go over in great detail in our course, and you will go over it in great detail in AMP2. Okay, in AMP2, you will go over the chemistry of ATP production. So it's something to look forward to in the future. But at any rate, hopefully you at least recall a basic description of aerobic cellular respiration from chapter 3. Okay, aerobic cellular respiration refers to the chemistry of making ATP from the food you consume while oxygen is present. Okay, while oxygen is present. This process of making ATP is fairly involved from a chemical perspective, and as I said, you will get this in AMP2. But some of the short take-home notes that I would want you to appreciate at this time is that aerobic cellular respiration is very efficient. Okay, it's very efficient. And what I mean by that is that for each molecule of well, let me put it this way. Each molecule of glucose that you send through the aerobic cellular respiration chemistry results in a production of 36 net ATP molecules. That is a whole bunch of biochemical energy. Okay, so each molecule of glucose, now of course glucose would come from your food. Glucose would come from your food. Each molecule of glucose that is processed by the chemistry of aerobic cellular respiration produces 36 net ATP. Okay, that means the profit margin on making energy is 36 molecules of ATP. That is very efficient. Okay. Most of the ATP that is made is in fact made in the mitochondria, as Lindsay mentioned in her answer on the chat window. Okay. Okay, so it's made in the mitochondria, then the ATP floats out of the mitochondria and can be used by cellular processes, such as regulating the contraction cycle of skeletal muscle cells. Okay, so ATP is produced through aerobic cellular respiration. Okay, and when a person has a need for a long expenditure of energy, such as you're running a marathon or doing some kind of sustained exercise, most of the energy that you're going to get is going to be from the ATP that's made through aerobic cellular respiration. Okay, so when you look at figure 10.11 on page 313, you notice that figure 10.11 highlights 3 different ways that ATP can be provided to muscle cells to fuel their mechanical motion. And one of them is letter C there at the bottom, ATP from aerobic respiration. Notice that ATP from aerobic cellular respiration can be used to provide energy that is sustainable over several minutes to several hours. Okay, so anytime your body needs a sustainable source of energy, it's going to try very heavily to rely on aerobic cellular respiration. Okay. An alternative to aerobic cellular respiration is what picture B on page 313 is trying to share with you. Your book refers to it as anaerobic glycolysis, and that's fine. You can learn it that way. Many other textbooks will describe section B as fermentation, okay? So anaerobic anaerobic glycolysis, which is also known as fermentation, okay? This process can provide energy for, as your book says, about 2 minutes before it kind of peters out and is not useful anymore. Can provide energy for up to 2 minutes. Okay. It is not nearly as efficient. It is not nearly as efficient as aerobic cellular respiration. It's actually quite inefficient. Excuse me. And there's nice allergies happening here today. Inefficient compared to aerobic cellular respiration. Anaerobic glycolysis, also known as fermentation, will make 2 net ATP for each glucose processed. Okay? So, if you were exercising for a long period of time, and your body's storage of carbohydrate that it could burn through aerobic cellular respiration started to become limited, or the amount of oxygen available to you because you're exhausting yourself through exercise, you're taking on an oxygen debt in your tissue. If that oxygen becomes limited, then the chemistry that your cell is capable of performing to make ATP also starts to become limited. And if the oxygen is not available, your cells are going to switch gears, so to speak, and go into this fermentation pathway where you only make 2 net ATP for each glucose. It's much less efficient, but it will keep you running for another couple of minutes. Okay? All cells in your body do this. Okay, so number one, ATP production in all cells of the body do this. Okay. Now, what's very unique about muscle cells is that they have the ability to do a third way of generating ATP rapidly in quick microbursts, if you will, using creatine. Okay, so let's see. A third way of producing ATP is unique to muscle cells. Okay, a third way of producing ATP is unique to muscle cells. And that is simply to create ATP from removing a phosphate group from a molecule called creatine phosphate. Okay, so let's put it down this way. During times of muscle relaxation, when your muscles are not fatigued, any excess ATP in the muscle cell is used to attach a phosphate atom to a molecule called creatine. Okay, during times of muscle relaxation, excess ATP is used to add a phosphate atom I see your question, Jennifer. I'll answer that in just a moment. During times of muscle relaxation, excess ATP is used to add a phosphate atom to a substance, to a molecule called creatine, okay? Creatine is a small molecule that resembles an amino acid. It is not an amino acid, but it resembles an amino acid, okay? Now let's put it this way. Come on. Creatine is a small molecule that resembles an amino acid, okay? And so what I'd like you to do is think about creatine as an uncharged battery. Think of creatine as an uncharged battery, okay? And what I'm saying here is that during letter A here of note number 2, 2A, is that during times of excess ATP production in the cell, if the ATP is not immediately needed, what the cell can do is take some of the energy of the ATP and use it to charge a battery, charge creatine by adding a phosphate atom to it, okay? To add the phosphate atom, you have to add a phosphate bond, and phosphate bonds, as you know, are high energy bonds. There's a lot of energy in a bond that attaches phosphate to a molecule, okay? So what we're saying here is that in times of excess ATP, the excess ATP will be used to charge a battery that we're going to call creatine. Okay? So, when your body needs quick bursts of energy, such as you're responding reflexively to a stimulus, and you just need to rapidly move a body part, okay? What can happen is your cells can mobilize energy very quickly by removing the phosphate group from the charged battery, okay? So during times where energy is needed rapidly in short bursts, and as your book says, about 15 seconds. Okay, during times when energy is needed rapidly in short bursts, the creatine phosphate can be broken down to create energy rapidly. The creatine phosphate can be broken down. The phosphate can be reattached to the ADP to form ATP, and then that ATP is immediately available for a cell to do something with it. Okay, during time when energy is needed rapidly in short bursts, the phosphate can be removed from creatine, transferred to ATP, transferred to ADP to form ATP, which is then accessible to muscles to power contraction. Okay, so this is a third way of providing rapid energy availability to a cell. Okay, Jennifer had a question on the chat window. Doesn't one glucose make 38 ATP? No, it doesn't. You might be getting confused about, well, let's put it this way. Different cells that you may study in biology do use glucose to get different energy yields. For example, bacteria, prokaryotic cells like E. coli, they do get 38 ATP per glucose. But animal cells, eukaryotic cells like the cells of our own body, only make 36 ATP. So Jennifer, there are some cells on the planet that do make 38, such as bacteria, but our own cells only make 36. Does that make sense? Okay, I hope it does. All right, gang. Questions right now. Okay. So in terms of providing energy for skeletal muscle contraction, if we need sustained energy over many, many hours of effort, we're going to use aerobic cellular respiration. That's the most efficient way. If oxygen becomes limiting, your tissue is going to start fermenting, and that's going to create a little bit of energy for a few minutes maximum. And then if we need short bursts of energy to power things for very brief moments of time, typically the skeletal muscle cell will rely on its batteries, on its creatine phosphate, to form the ATP and rapidly use that for whatever process is needed over a period of up to maybe 15 seconds. Okay. All right. Gang, any questions at this time? Okay. All right. So let's talk, if there's no questions about this, let's talk about muscular dystrophy. Okay, muscular dystrophy obviously is a muscle disease. Okay. So muscular dystrophy is a single disease that has multiple different causes. Okay. So muscular dystrophy is a disease that's caused by a problem with the dystrophin protein. I've hinted about this in the past, and I'll mention it again. For example, on table 10.2, page 302, that table lists the dystrophin protein as a structural protein. Okay, so it's in table 10.2 on page 302. And dystrophin as a protein is graphically illustrated on page 299, picture D there. You can see it's kind of drawn as a light blue rectangle. Okay, so muscular dystrophy is a single disease that has multiple different causes. All forms of the disease involve abnormalities in the dystrophin protein. And again, I'll reference you to page 299 so that you can look at it. I mean, it doesn't really show you the structure, but it shows you symbolically what it looks like. All forms of disease involve abnormalities in the dystrophin protein. You know that the dystrophin protein is responsible for tethering the myofibril to the sarcolemma. In other words, dystrophin acts as a bridge between the myofibril and the sarcolemma. Okay. Whenever the myofibril shortens, the tension of shortening the myofibril has to be transmitted to the sarcolemma to allow the actual cell to shorten. It's dystrophin that actually transmits the tension of myofibril shortening to the muscle cell plasma membrane so that the cell itself can actually get shorter. Dystrophin transmits the tension of myofibril contraction to the sarcolemma. Okay. Now, bullet point number 1 says it's a single disease that has multiple causes. Gang, the multiple causes is the realization that different mutations have been identified in the dystrophin gene. Okay. Scientists have identified over 50 different mutations in the dystrophin gene. Each one of these different mutations leads to a different abnormality in the dystrophin protein. Okay, so all forms of muscular dystrophy involve an abnormal dystrophin protein, but that dystrophin protein can be messed up in over 50 different ways. Okay. The end result of having an abnormal dystrophin protein is that typically the tension from myofibril contraction is not successfully transmitted to the sarcolemma properly, and so the muscle fibers may not shorten at all, or they may in fact over shorten and actually tear the muscle fiber apart. Muscular dystrophy typically results in physical tearing of muscle cells. Of course, the tearing of the muscle cell leads to the death of the muscle cell given enough time. Over time, this leads to death of muscle cells. And of course, you know, if you just have a couple muscle cells dying, that's not too big of a deal, but over time that's going to lead to traumatic injury to the body. If you have muscle cells dying in your heart may become so weak that your heart really cannot successfully push oxygenated blood through your body, and you basically suffocate. Okay, so this is a really bad disease. I mean, it's something that you don't wish on anybody to have to experience.
I have a question.
Yes.
Muscular dystrophy, it shortens up your muscles. What happens if your muscles are like overstretched and can't contract to the normal state that it was in?
Overstretched and cannot contract.
Like,
Well,
say you were over 600 pounds, but then you can't have your muscles come back to the way that they were if you lost a lot of weight.
I see. Well, that's a complicated question because as long as there's no genetic abnormality, the muscle fibers should be able to repair themselves given enough time. But if you're not able to really bring your muscle tissues back to their normal length, that's likely because you have too much connective tissue. Connective tissue tends to be fairly avascular. There's not a very rich blood supply. So when you have connective tissue, it doesn't remodel itself very rapidly. Okay, other questions? Okay. Now, coming back to muscular dystrophy here, muscular dystrophy is found predominantly in males. It can happen in gals, but it's much more of a male disease. Most commonly impacts males, although cases sometimes occur in females. And the reason it impacts males most predominantly is because the gene for dystrophin is actually found on the X chromosome. The gene for dystrophin is actually found on the X chromosome. Now, you might look at that at face value and think, well, that's kind of odd because the X chromosome is sort of the female chromosome. So if it's on the female chromosome, why doesn't it impact females more? And the answer is very simple. Remember that all of our cells are in fact diploid. That means we have 2 copies of every chromosome. You have 2 copies of chromosome number 5. One you got from dad, one you got from mom. Okay.
What can happen if you only have 1 of each and not 2?
Well, let me see if I address your question in just a moment, Suzette, because that's sort of the foundation here for muscular dystrophy and how it impacts males more frequently than females. Our cells are diploid. Each cell has 2 copies of each chromosome. You have chromosome number 5 from dad and chromosome number 5 from mom, right? And it's the same thing for all your chromosomes. You have chromosome number 17 from mom. You have chromosome number 17 from dad, okay? Where the difference comes into play, however, is with the sex chromosomes, right? Males have XY sex chromosomes. Females have XX sex chromosomes. All right. So the reason that this disease predominantly impacts males is because if you have a mutated dystrophin gene on the X chromosome in a male cell, males only have 1 copy of that X chromosome, which means they have the defective gene. That means they're going to have the disease. But with gals, if you have 1 X chromosome with a defective gene, you can very easily have the other chromosome have a normal, healthy copy of the gene, and that is enough to rescue and prevent that woman from having the disease. So as long as you have 1 copy of a correct gene on one of the X chromosomes in women, then gals typically don't suffer the consequences of the disease. It's only when gals are unlucky enough to have 2 mutated copies of the dystrophin gene on each of the 2 X chromosomes that the gals end up with the muscular dystrophy disease. Okay. Because males have only a single X chromosome, if the X chromosome contains a mutated dystrophin gene, the male will experience the disease. Because females have 2 X chromosomes, I'll learn to spell here. As long as one of the chromosomes has a normal dystrophin gene, the female is spared, the female is prevented or rescued from having the disease. So to answer your question again, Suzette, in general, if you have, in general, human disease is extremely complicated and it's very difficult to make an umbrella statement that will be true in all circumstances. But as a starting point for the conversation, if you have a disease that's caused by a mutated gene, as long as you have one good copy of the gene on one chromosome and a mutated copy on the second chromosome, usually the severity of the disease is much, much less. But if you have two copies of the mutated gene on both chromosomes, then the disease is quite severe. Suzette, did I answer your question?
Yes, you did.
Okay. All right. Jennifer, is muscular dystrophy sex-linked? Yes, it is, Jennifer, because it's found on the X chromosome. Yes. All right. Gang, do you have any questions about muscular dystrophy, or does it make sense, the genetics of it? What I'm telling you is that males are impacted more frequently than females. It's because men only have one copy of the X chromosome. So if their gene on that X chromosome is mutated, they are going to have the disease.
What's the percentage of a male getting it? Is it like 100% like that he's gonna get it, or is it like a 50-50 chance he might get it?
Well, I don't know the statistics in our population off the top of my head, but if a male does have a mutated dystrophin gene on his X chromosome, he will have the disease. Now, as I mentioned, there's 50 different versions of this. There's over 50 different abnormalities that have been documented by medical science. So some versions of the mutation produce worse disease than other versions of the mutation. Lindsay asks in the chat window, so is a female, if a female has two mutated X chromosomes, that is when she has the disease? Yes, Lindsay, that is when she has the disease. Okay, other questions, gang? Okay, so, you know, when I say we, I mean medical science is known about the problem with muscular dystrophy for probably at least 50 years. We've known about the issue with muscular dystrophy for 50 years, and yet we still do not have a cure for it. And that's because even though we know what's wrong, the technology to actually correct the problem is still not good enough to actually fix it. Okay? Getting, you know, the future of our medical care system includes gene therapy, the idea that we can correct defects in our genes. But doing that is extremely difficult. And the technology to do that safely and efficiently is still, well, still a matter of research. We just can't do it. We know what's wrong, but we don't have the technology yet to fix it. All right. Gang, any questions about chapter 10 today? Okay, folks, what I'm going to do at this time, it is 9:14 by my clock. I'm going to stop for the day. And you can expect that on Monday, I will start chapter 12, which is the nervous system. But the first thing I'll do on Monday, the first thing I will do on Monday is ask you if you have any questions about your upcoming exam. Okay, so Monday we have normal lecture. We will start chapter 12, but I will ask if you have any questions to prepare for your lecture exam, which is next Wednesday, the 15th.
Question. Will we have any like extra, like with our last lab exam, you had that paper for us to know all the anatomy in the female or whatever that was. Are we going to have something like that again?
I'm not sure of your question. Are you referring to the anatomical position, woman?
Yeah. Are we going to have something, another paper we need to print out along with the lab exam or the lecture exam? Or are we just simply studying our notes for that exam?
Okay. For everyone in the meeting right now, please type into the chat window your answer to my next question. Did you receive my study guide? Okay. So the study guide for lecture was sent out, and that included everything you would have needed to know. So is there confusion about this? There's not going to be another anatomical woman chart on the exam, but there will be a matching activity where I give you statements about the different forms of joint motion terms like circumduction, pronation, these kinds of terms, and you have to match the descriptive phrase to the proper motion term. I also, okay, another question coming through the chat window here. Okay, my question through the chat window. Did you receive the joint motion matching practice sheet? Please type in, okay, good, you did. So all of these things, right, the study guide describing what's on lecture exam 2 and the joint motion matching practice sheet have been sent out through email. So if you didn't get those, first I would ask you to please check your email again, and please check your spam folder, your junk folder. And if you still haven't gotten these things, please send me an email and I will send them to you directly. Okay, Lindsay asked, will we bring a note card to the exam as well? Well, you know, in the funny times we live in right now, you're going to be taking your exam at home, and there's no way I can stop you from having a note card. There's no way I can stop you from having your textbook and notes out. So I'm not even going to try to do that. That would be silly. You can use whatever materials you want, but keep in mind that the exam will be timed. Okay, the exam will be timed at 1 hour and 25 minutes, as I would typically give you face-to-face in class together if we were at HAC. So you can use your resources, but you only have 1 hour and 25 minutes to do a good job on the exam.
Dr. Allen, I have a question. For the upcoming lab exam number 3, which is on the 4th, are you going to be giving us a paper soon, being that it's a month away?
I will be. You will be getting your next word bank next week, when we would have theoretically been coming back to lab for a normal activity. So expect to get that next week. Pam has a question. Will the contraction cycle be included in the essay? Pam, not in a formal way, but you can expect that I may have questions about the contraction cycle sprinkled throughout the exam itself. Anything else at this time before we close up shop for the day? Okay, will we have lecture on Wednesday? The Wednesday of the lecture exam, there will not be lecture. There will be no lecture on the Wednesday of the exam. Okay. And Pam asked about no lab. What, Pam, do you mean no lab today or no lab next week? Today. Hold on one moment. Yes, there is no lab in a formal sense today. I will remain online in about 15 minutes. I'll be online. If anybody needs my attention, needs my help, wants to talk about something, I will be online in another 12 to 15 minutes. Jennifer asks, is the test from 8 to 9:15? The exam will be anytime on Monday. Excuse me, the exam is next Wednesday. The exam will be anytime between 7 a.m. and midnight. You can take the exam anytime between 7 a.m. and midnight. Okay, anything else? Okay, folks. So I'm going to stop sharing my screen with you here. And I'm going to go take a quick break. If you don't need me, you're free to head out. Please have a nice weekend and be safe. Be conscientious about what you're doing and how you're potentially exposing yourself to the coronavirus. I'll be back online at 9:35 for anybody that has a need to spend more one-on-one time with me, that I can help you. Okay? So until then, I'll see you soon, or have a good weekend. I'll be back online at 9:35.
Have a good Easter weekend.
Thank you. You too.
Well, that's what he needs to do.